So next up we have James Logan, who's Head of Department of Disease Control at the London School of Hygiene and Tropical Medicine. His research interests include investigating new ways to detect and control diseases, including COVID, malaria, Zika, dengue, trachoma, and Lyme disease. Most recently, James is leading a study with funding from the UK government to investigate novel non-invasive diagnostics for COVID based on VOCs and to determine whether dogs can be trained to diagnose COVID-19 infection in humans. Thanks, James, and over to you.
Good afternoon, everybody. First of all, thanks very much to the organisers for inviting me to present our work today on training dogs to detect the odour of COVID-19. This is a collaborative effort. It's been led by myself at the London School of Hygiene and Tropical Medicine, but collaboratively with Claire Guest from Medical Detection Dogs, which is a UK charity which specialises in training dogs to detect medical conditions, and Professor Steve Lindsay at the University of Durham as well. Now, you will have heard some of this sort of rationale behind the need for different types of testings in previous talks, I'm sure. Very briefly, currently COVID testing is pretty labour intensive, it's invasive, and it's pretty slow. And although there are some things in the pipeline that could speed up that process and be a little bit less intensive, they're certainly not there yet. There are also temperature checks which are being done at places like airports and schools, for example. But when you look at the data for that, it's not particularly scientifically robust. So there is a real need for a rapid screen of particularly asymptomatic individuals for COVID-19. So back in January, we sort of had the idea of doing this. And of course, at the time, we didn't really know whether COVID-19 created a specific odour. I think now, based on what I've seen with our own work and with what's been done elsewhere and what we've even seen today, I think we can be pretty sure that there is an odour associated with the infection. Whether that's specific or not is another question. But there does appear to be an odour there. Now, I would have really loved to show you our results today, but unfortunately I can't, because we haven't quite progressed to where I thought we would have by this point. And the reason for that really is that when we started this study, which was just as we entered lockdown, the first lockdown, the number of cases in the UK dropped dramatically, which meant that we really struggled to collect enough samples to do the work. So we have been collecting over the summer, and we're pretty much there now, and obviously that collection has improved somewhat dramatically within the last few weeks and months. So what we'll do today is I'll give you an overview of what it is we're actually doing with the dogs. And what I can say is that the results so far look very promising. I'm also going to tell you about what led us to this point as well, to think about using dogs in this way. So I'm going to tell you about some of the previous work that we've done with medical detection dogs on a different disease. So if you'll forgive me, the talk that I'm giving isn't all about breath, and it isn't all about COVID-19, but hopefully you'll find it interesting nonetheless. So the disease that we have worked on most recently and published recently with medical detection dogs was looking at malaria. And as I'm sure you know, malaria is transmitted by a mosquito. And the parasite spends some of its time inside the mosquito and some of its time inside us. And we wanted to know whether having an infection would change that transmission cycle of the mosquito. And what we demonstrated was that when the mosquito has a malaria parasite inside it, it changes its behavior. It actually becomes more attracted to human odor. So the olfactory receptors are changed with the mosquito when it has the infection. And interestingly, it's only at the later stage of infection, when the sporozoites are present, when it is infectious, that this behavior occurs. One question we had was, does this happen in humans? And we had anecdotal evidence that when people were infected with malaria, they became more attractive to mosquitoes, seemingly. Now, there's a real link here, because the way that mosquitoes find us is through odor. So it's likely that if this is happening, there would be an odor link there. And again, what you might expect if this is being driven by the parasite from an evolutionary point of view is that at the early stage of infection, people might be less attractive because the parasite doesn't want to be transmitted at that stage. But at the later stage of infection, they would be more attractive because that's when they can be transmitted. Now, this sort of phenomenon where pathogens manipulate their host is really prevalent throughout the animal kingdom. We have many examples where this happens. This is one of my favorite ones, where basically an ant gets infested by a fungus. That fungus brainwashes the ant. It causes it to climb up a stem so that it's high up in the air. The fungus then grows out of its body that you can see here, and then it sporulates, and the spores then disperse much further across the ground than they would if the ant was on the ground. And there are lots of examples like this. So in the study that we did, we wanted to know, was this happening with malaria when people are infected? So in this study, which was collaborative with Rationale University in the Netherlands, ISIPI in Kenya, and also the MRC unit in the Gambia, we looked at this phenomenon with malaria. And we did it in children, and what we did first of all was we got participants to lie inside tents, and the odor from the tent was blown into a chamber which contained mosquitoes, and we could measure the behavioral response of the mosquitoes to the odor coming from the chambers with people sleeping in there. Those people were then tested for malaria to see whether they were infected or not. So what we found was that people who were parasite-free, so this is the level of mosquito response to those individuals, then at the different stages of infection, starting with the early stage infection, moving to the later stage, and the final infectious stage, you can see that people became significantly more attractive at that point. And that was odor-mediated. It was the odor being blown from the tents to the mosquitoes. The same individuals were then treated with an antimalarial, and what you can see is after the treatment, they were that sort of pattern in change in attractiveness was no longer there. So it was likely to be due to the infection and not just due to individual differences in those groups of individuals that we looked at. What we did next was collect their volatiles, and we know that foot odor in particular, it's particularly smelly, but also it's a very good stimulus for mosquitoes. And so and you probably know that yourself, if you've been bitten around your feet or your ankles, it's probably why. And that's why you should wash your feet often as well. It's a good incentive. But what you can see here is we used air entrainment to collect the headspace from around the foot onto Pluripac and Tenax filters, which we could then elute and analyze. So we can analyze these by GC and by GCMS, but we can also use another type of sensor. So we can connect microelectrodes to the antenna of a mosquito. And you can see here a mosquito which has been decapitated. The cells inside the mosquito stay alive. We can connect the electrode into one end of the head and tuck the antennae into the other electrode, which creates a circuit. And we can then measure the cumulative response of the receptors on the antenna of the mosquito. And in that way, we can tell and we can see what a mosquito is smelling. So this is an example of what we get. So in the top trace, that's the GC trace of the individual compounds, and the bottom trace is the response of the mosquito's antenna. So where we get a depolarization in the response of the olfactory receptors, matching with a peak on the GC, we know that the mosquito is able to detect that specific compound that can then be identified. So by using this approach and also some multivariate statistics, we were able to pull out individual compounds that seem to signify a malaria infection. And it's quite a complicated graph here, but the take-home message really is that there appear to be compounds, particularly aldehydes, associated with the infection. And this compound down here, 2-octanone, which seems to be very much associated with gametocytes, which is the very late stage of infection when people are infectious. The compounds themselves, the pattern changes depending on level of parasitemia, so how big the infection is, the parasite load. as well. So we can use this specific pattern of ratios of these particular compounds to signify a malaria infection at different stages of infection. And then some of the other work that we've done, which I don't have here to show you, is that we can actually distinguish between different strains of malaria parasites of the same species based on the odor profile that's produced during infection. So following on from this work, after we identified the compounds, we took it back to the mosquitoes to tell us whether we got it right. And we used traps on the Bijagos Islands, which you can see here, a beautiful place in West Africa. And we were able to demonstrate that when you add these malaria compounds to a trap, you can significantly increase the trap catch of malaria mosquitoes, and in fact, other species of mosquito as well. So that gave us confidence that we had the correct compounds that the malaria mosquitoes certainly were detecting. Now, this work is continuing. We have funding, and we're working with the Bill and Melinda Gates Foundation to develop a sensor for the detection of asymptomatic malaria. And that's very important. All of the participants in that study were asymptomatic. And that's one of the biggest problems when trying to get rid of malaria, is to find those people who are asymptomatic and responsible for residual transmission and importation of malaria into countries where elimination has been achieved. So we are actually working with a variety of organizations, and we're still looking for partners. So if there's anybody out there who works on the development of sensors for VOCs and would like to talk to us, then please do get in touch. We'd love to hear from you. The other thing that we're doing on that study is finding a pathway through the World Health Organization for approval and procurement of a non-invasive volatile odor-based diagnostic for malaria as well. So there's a big piece of work to be done there. Now, we'd love to imagine, taking a mosquito out on a lead and getting them to sniff people to tell us whether people are infected. But and until we have an electronic sensor that can do it, we turn to our colleagues at Medical Detection Dogs, because dogs are amazing creatures, as I'm sure you've heard before. They're one of nature's greatest detectives. They've got an incredible sense of smell. They can detect, a teaspoon of sugar in 2 Olympic-sized swimming pools. It's that sort of sensitive, parts per trillion. But not only can they detect stuff, they can be trained very easily. Now, Medical Detection Dogs, the charity that we work with, is world leading in this field, and really sort of was instrumental in getting the initial robust data behind demonstrating how well dogs can actually do this. And there's plenty of scientifically robust, peer-reviewed papers demonstrating that dogs can detect various medical conditions, including different types of cancers, changes in blood sugar levels, oncoming seizures, the presence of Pseudomonas bacteria, and with us, malaria, which I'll tell you about now. So for this part of the study, we asked our participants in the Gambia to wear nylon stockings on the feet. Nylon's an extremely good matrix for collecting odors, and we collected those socks essentially and put them into pots, which were then shipped back to the UK. Now, for the training of the dogs, you can see that the pot is here in this metal stand, and there's a grid across here, so the dog never comes into contact with the odor, with the sample itself, but can smell the odor coming from the sample. And there are a number of these stands. The dog's asked to walk down the stand. It's been trained to sniff each pot very briefly, and when the dog signals correctly on the correct pot, it is given a reward. So it's given its favorite toy or a food reward. And so in the same way that you would train a dog to sit or come or some other command, we do the same for training them to detect odors. And it's very robust, and you can see that the response is very definite. So the dogs, it's a very passive response. It's not barking and jumping. It's sitting or pointing and staring with their nose. So I'm going to show you a short video here, I hope it works, of the dogs detecting the malaria samples. So you'll see this dog here, which I think is Lexi, moving down the different samples, having a sniff. And the malaria sample was in standing on the floor. You can see the dog stood and told the handler that she was detecting something there with a very definite response. And you'll see how definite this response was looking at it from this angle here. So in the first three pots, these are controls. They're from people who were uninfected. And there you can see the response of the dog to the malaria odor. So you can see how very sort of definite that response is. And the charity is also working on the development of a way of determining how great the pressure is of the dog's nose when it actually detects that odor. What we're working on now is training the dogs to detect from a distance so that the dogs aren't sort of pressing right up against it. So that work is also continuing, and we're particularly looking at using dogs at ports of entry, specifically at border crossings where lots of people need to be screened very quickly to stop cases coming back into communities when malaria cases are low. The dogs are reaching, so 81% sensitivity, 92% specificity, but actually when you look at the sensitivity data based on the WHO cutoff for diagnostic, actually it's above 90%. So these dogs can do this with really good accuracy. So following on from that work, which we published last year, as I said, back in January, we thought, okay, there are problems with COVID-19 diagnostics. What can we do here with dogs? And I remember sending a text to Claire saying, COVID dogs, question mark, what do you reckon? Crazy? And she said, no, not crazy at all. Let's give it a go. So we embarked upon this study, excuse the pun, and pulled together the same team of people and dogs, in fact, for the initial work. And as I said, it's collaborative between the school, Durham, and medical detection dogs, but also Cardiff University, who's helping us with some of the identification work by mass spec. And it's funded by the UK government, the Department of Health and Social Care. It took a bit of a while for us to obtain funding, but we've managed to get funding to fund the study, which is great. And at the moment, we have 20 dogs in training, and these are some of the dogs that you can see here who are currently in training in phase 1. So for this particular study, we aimed to collect, based on our sample size calculations, we needed 325 positive people and 675 negatives. Now, at one point over the summer, it looked like we might have to recruit around 16,000 people to get enough samples because of the difficulty with the cases being so low. However, that luckily wasn't the case, and we've pretty much hit our target now. We have 24 NHS hospital sites that are recruiting, and they're recruiting asymptomatic and mildly symptomatic staff and patients as well. And we're also recruiting members of the public, which are being recruited through advertising at drive-through testing centres and also at universities. And we have set up a call centre at the school to help us with that, for people to call in and recruit them that way. And then the samples are collected at home. We send them a pack. The packs are sent back to us to then process the samples. We're collecting in three different ways. So, we need to make it as simple as possible for the participants, because a lot of them are having to do the collection themselves. So we're collecting on face masks. So people are being asked to wear face masks for three hours. We're also collecting on shirts and t-shirts and nylon socks as well. And those are being worn for 12 hours. Once the samples are sent back to us, at the school, we process the samples. We extract them. We're doing solvent extraction. We're also doing air entrainment and headspace analysis, and analysing by GC and GCMS, as I say. And that work is ongoing, although we do have some interesting initial results from that. And we're doing that collaboratively with Professor John Pickett at Cardiff University. The samples are being grouped as people who are negative and positive, and then processed for use in the dog training. But we are looking at other factors like severity of symptoms. So we've got samples from people who are asymptomatic, people who are pre-symptomatic, so they didn't have any symptoms, then they went on to develop symptoms. And it'd be interesting to see whether dogs can tell the difference between somebody who perhaps didn't ever have symptoms, somebody who had no symptoms and then had mild symptoms, and somebody who went on to develop severe symptoms. And we have samples representing those groupings, and also from different demographics, etc. as well. So we'll be teasing that apart to see whether there are any odor differences in those different cohorts, and whether the dogs can detect those differences as well. Also, we're looking at working with partners for sensor development as well. We have obviously we have a large clinical trial set up for the collection of samples, which is going very well, and there's an opportunity there. And there's also opportunity to use some of the samples that we've already collected, where perhaps the headspace of those samples or extractions of those samples could aid in the development of electronic sensors. So again, we're very, very keen to speak to any organizations. We're already speaking to some, but if anybody's interested in that, please do get in touch. So for the training of the dogs for this particular study, we're doing it in 3 different phases. So in the first phase, the dogs are being trained using the stand system that you saw previously. There is a period of training where the dogs are basically given access to infected samples, and then also given access to a lot of uninfected samples. And it's very important that the dogs learn to ignore the background. So they learn to ignore people who do not have COVID, but might have something else, or might just smell a bit differently, different households or different hospitals, etc. So the controls are almost as important as the infected samples. We are performing a double-blind randomized trials to work out the level of sensitivity and specificity, and we will know that by the end of phase 1, and we're just sort of approaching that now. Then we will move to phase 2 and phase 3. Phase 2 is sort of a scale up from a laboratory environment to a real world situation. So instead of dogs detecting on samples, we want dogs to be detecting actual people. Phase 2 has actually already begun. So we have a bank of dogs who are now trained to detect odors from people and from whole odors from people, as opposed to an individual bit of sock within a pot. And so they are now ready to go. As soon as we've got the first phase 1 data, that training will begin. Within a few weeks, those dogs will be ready to be deployed. So where could we use dogs? So, each dog, in theory, could screen up to 250 people per hour, which is really quite remarkable. It is very much dependent on the location and what's feasible within each location. One of the primary places that we're looking to deploy is airports, and we're talking to various airports at the moment and have plans to do some trials there very soon. There are different ways that we might use dogs in airports. One might be to use them at security or even at check-in to make air travel safe and give people confidence to travel. The other way would be to have people screened as they come back into a country, particularly from hotspot countries, before coming in, and then they'd be asked to isolate, etc. But there's a lot of scope to use dogs in other areas as well, including sporting venues, anywhere where there's a lot of people that have to be screened very quickly. So we're talking to various organisations about how we might be able to deploy in those other circumstances as well. One of the useful things about this study and working with the government is that not only have they provided funding and support, but we're able to talk to them about how we might scale up and deploy. And there's a task force that has been set up specifically for this purpose, and we're in discussion about how we can get the dogs out, where will be most appropriate, considering the timing of the pandemic, etc., etc. We're also talking to different dog training agencies in the military and also border force and the police about how we will scale up as well, because we will need a number of dogs to be able to do this effectively as things start to reopen, and we need to see how feasible that will be. So, and the beauty of this also is that dogs can be deployed very quickly. So within 8 to 10 weeks after training, they can be deployed. But there's a bit of work still to be done in terms of demonstrating how well the dogs can do this, and working out at the time when we deploy, where are we in the pandemic and where are the most appropriate places for dogs. We absolutely do not anticipate dogs being the answer to everything. This will be, if it works, this will be a rapid screen for situations where we have to screen a lot of people very, very quickly. So we're extremely hopeful that this will be good. So please watch this space. As I say, I don't have results to show you other than to say the results look very, very promising. And so we're hoping that we'll we don't want to jump the gun. So we're hoping that we'll have those results pretty soon to be able to share. And as I say, we do have this setup running at the moment, and we're very, very happy and keen to collaborate with anybody. So do get in touch. So I'll finish there, and just to thank all the different people who are involved in this. These are sort of the main people involved in the trial. But of course, we do have our 24 hospital sites and all the Pillar 2 remote testing sites up and down the country as well, helping us with this study. So that's well over 100 nurses and doctors and various people. And we're very grateful to their help, for their help. So thank you very much.
Brilliant. Thanks so much, James. That was fantastic. So lots of questions coming through. I thought I could kick us off with 2 quick ones. So you mentioned there's some of the lab-based work being done as well. So is the hope that if the dogs do work, you'll also have a defined identification of some of those VOCs of interest from the lab-based work? Is that the hope?
Yeah, so I mean, we have the GC and GCMS work going on in parallel. So we're looking at the identifications that we've got there already, and able to compare. Obviously, we've got negative people, we've got positive people, and we've got people who are negative with symptoms of other infections as well. So we're able to sort of tease apart looking at the volatiles there. The dogs are also extremely useful because they can tell, if they're detecting it, they've learned the smell. We're also working on pseudo odors, so basically, making a synthetic mixture of what COVID smells like, and the dogs will tell us if we've got that right or not. So they're a really useful aid, research aid, when you're looking at identifications and even the development of sensors as well.
On the malaria work, so you have VOCs which are signaling effectively. So on the one hand, the more volatile it is, the better, because the mosquito is going to come from further away to the person. But with the sampling that you were doing with people wearing socks, for instance, it seems that the compounds are likely to be less volatile. So in what you were looking at there, could you just comment on, was it the very volatile end, or was it less volatile, semi-volatile type compounds?
Yeah, so actually, when you use nylon as a matrix for collecting volatiles, you actually pick up a lot of the volatile stuff as well. You're not talking about the very, very low molecular weight compounds, but certainly low enough to sort of C7, and those are the compounds that mosquitoes tend to respond to. So the nylon is very good at that. Actually, we also use it as a matrix to create synthetic odors that we put out as well with those volatile compounds. It releases them sort of steadily over time as well. But for some of the malaria work, we also did air entrainment of the foot itself. So we're able to compare what we're seeing on a sock versus what we see coming from the skin itself. It's pretty comparable, really.
So is dogs' ability to recalibrate its sense of smell a challenge? Can they experience olfactory fatigue? As in, when we go to perfume shops and smell several different perfumes, we smell a bit of coffee to reset our sense of smell.
Yeah, absolutely. I think that sort of thing can happen. I think in a real world sort of deployment situation, we have a slightly different problem, and that is that dogs are going to be smelling a lot of people who are not infected, with the odd one or two who is infected. And that's a challenge, because there's a chance actually they might forget the smell of COVID. and start to learn something else. So throughout the training, it's very important, and throughout deployment, the validation and reinforcement of that training is really important. So for example, when the dogs are deployed, what has to happen every so often is a dummy person goes into the mix. So a plane and people come off, for example, and somebody gets put in there, somebody from the team, obviously, with the pseudo order on them. So we know the dog is still picking up on that person, and they haven't forgotten the smell.
Is there a risk of infection or viral transmission of biological sample with COVID-19 towards the dog during training?
Yeah, I mean, it's a very good question, and something we've been extremely cautious about, and have taken a lot of considerations around that. So when you look at the scientific evidence, dogs, there is evidence, particularly from a study in Italy, demonstrating that some dogs in households that have people who are infected with COVID-19 also become infected. So you can find the virus in the dog. There's little to no evidence that the dogs become sick, and there's no evidence that the dogs onwardly transmit. So they don't transmit to other dogs, and there's no evidence they transmit onwards to people. So although, so the risk is very low generally, but of course, we are considering that, and we have protocols in place, obviously, to protect both the handlers of the dogs and the dogs themselves. The welfare is really important. And so we have protocols in place that if a dog becomes contaminated in any way, that we can decontaminate the dog with alcohol wipes. It would be taken out of operation. And also the dogs are regularly tested as well as the handlers. But the samples we're using don't contain active virus, so the risk is even lower then. Out in deployment, there's a risk that somebody, coughs on the dog or touches the dog when they shouldn't, at which point that dog would be taken out of operation for sure. But it's something we're monitoring. Obviously, we have vets on the committee who advise us on that.
What measures are undertaken to ensure sample integrity, stroke stability prior to presenting to the canine panel?
Sorry, can you say that again?
What measures are undertaken to ensure sample integrity and stability prior to presenting to your canine panel?
Yeah, it's a good question as well. So, I mean, we've got 20 years of experience of collecting volatiles in very similar ways, storing them, and doing the analysis. And we've done hours and hours and hours of work looking at the integrity of samples from the point of collection. So quite often we're in sub-Saharan Africa collecting samples from people in very hot conditions, and those samples have to be transported, and storage can be a problem, and all sorts of things. So we do have a method which includes cold storage, and particularly how the samples are sealed, what's collected in a usually under cleaned nitrogen or some sort of other environment where reactions are less likely to occur, and kept as cool as possible. But we have a strict and stringent procedure that we use to ensure that.
Time for one last question. So what extraction procedures used for extracting VOCs from the mask, which are then analyzed in GCMS? So I think the question is it thermal desorption, SPME fibers?
Yeah, so a mixture of things. We're thermal desorption is one, but extract, so air entrainment of the headspace around the mask, and then thermally desorbing the polymer is one method. And SPME is something that we might look at. We've not done that yet, but that's something we might look at as well.
Excellent. Okay, well, thanks so much. I think that's all we have time for in this session. I'm sure James